The results of the study were published in the New England Journal of Medicine. Researchers tested engineered replicas of the variant against blood samples taken from 15 people who had received both doses of the Pfizer-BioNTech vaccine during clinical trials. And what they discovered in those blood serum samples was a reduced neutralizing antibody effect, but still sufficient to neutralize the virus. A statement from the two companies says that while there is no clinical evidence to date that the 501Y.V2 variant escapes the protection offered by its vaccine, they are taking the necessary steps, including engaging regulators to develop and seek authorization for an updated mRNA vaccine booster once the strain that significantly reduces the protection from the vaccine is identified.
You would expect that with these mutants which are trying to escape from control, either by the immune system or other interventions, those kinds of mutations are something that you have to be concerned about and pay attention to. And what we're doing, Steve, is that we're making already, in case we have to use it to try and stay a step ahead of the game, versions of the current vaccines that have been upgraded to be able to be directed specifically against some of the mutants, particularly the mutant in South Africa, which is the most troublesome one.
Preliminary findings also point to reduced efficacy of other vaccines against the variant, including Moderna, which uses similar mRNA technology. Johnson & Johnson was found to be 57% effective in preventing moderate and severe symptoms, while Oxford-AstraZeneca was found to offer limited protection.
For countries that have reported the circulation of the SARS-CoV-2 variant, we recommend the acceleration of their preparedness to introduce all vaccines that have received emergency use authorization or approval by the regulatory authorities. Consideration should be given to the effectiveness of the vaccine against the variant, that is the N501V2 variant.
mRNA technology, a rapid process that instructs cells to produce a viral protein that triggers an immune response, can be adapted in existing vaccines.
What's going to happen, and this is inevitable, that this virus, which is an RNA virus, and RNA viruses tend to mutate readily. Not every mutation has a functional consequence, but some do, either by making it more transmissible, by making the virus more virulent, or by evading the vaccines. So what we have to do, when you ask me what keeps me up at night, is having a situation where we have a mutant that really escapes everything. I don't think that's the case because our technology is such that you could always make a vaccine pretty quickly with this advanced technology that you could address a given mutant. But it is concerning that you need to stay ahead of these mutants and essentially crush this outbreak so that there's no more replication. And when there's no more replication, you're not going to have any mutations.
Epidemiologists have noted the latest Pfizer lab study, but need to further extrapolate the vaccine's efficacy in real-world scenarios where the 501Y.V2 variant is prevalent. Sherman Bryceby's SABC News, New York.